The first step of the biomarker identification pipeline is to get your initial gene list. There are different ways to do it. One way is by doing a gene signature meta-analysis. In this case, the first step is to do a first filter of the gene signatures. You can select the secondary databases where to search and different filters, like for example, a keyword containing the name. Then Bioplat shows you all the gene signatures which pass the filters. You can select the gene signatures that will be included in the meta-analysis one by one, or you can include all of them. We can also include gene signatures that are open in your Bioplat desktop. Finally, you have to pick up the algorithm to apply to the gene signatures. In this example, we keep the genes that are at least in 50 of those gene signatures. Here we have the result, a new potential gene signature with 53 genes. In next steps of the pipeline, you can validate its prognosis value using different data sets, but also optimize it using feature selection algorithms and meta-heuristics. You can also get your initial gene list by data mining on the gene expression levels and some expression regulators, including microRNA, methylation, and copy number variations. For example, we can analyze correlation between gene expression of each gene and microRNA. The result of the process allows you to see correlation measures and survival plots. But, most important, you can use the result as a new Bioplat gene signature. You can also create a new blank gene signatures and add the genes you would like to work with. Datasets in Bioplats are useful for measuring the prognosis value of gene signatures, but also for training, testing, and validating new optimized models. Datasets are composed by expression and clinical data, including survival. You can import them from different sources, but then use them in exactly the same way despite the source you have used. One of the databases which Bioplats integrate with is the Cancer Genome Atlas TCGA. You can import datasets from there through a 4-steps wizard. In the first one, you pick up the dataset. In the second one, the case list name and the genetic profile. In the third one, the gene list to import. And finally, the clinical data, including survival. After importing, you can see the expression level and clinical data, edit its basic data or clinical data, remove samples, apply some arithmetic operations, filter genes, among other operations. The platform offers also a convenient operation to split randomly the original dataset into 3 new different ones for training, testing, and validation. We'll show you later. You can import datasets from your own files in geo format. After importing, you can work with it as we have done it with the previous one. You can also import datasets from our Bioplat cloud. It contains public samples and datasets curated by our scientific team. As well, it offers a web user interface that allows users and institutions to store privately their own molecular and clinical information, including genome, transcriptome, copy number variation, methylation, and microRNA profiles. They can also create datasets which includes public and also their own samples. Bioplat users can access the public and the private information from the Bioplat Cloud web application using the search menu. But also, they can import samples as datasets in Bioplat for gene signature validation and optimization. After importing, you have a new Bioplat experiment in your desktop to work with. Once you have the datasets and your potential gene signature, the third step of the pipeline is the statistic validation for measuring the prognosis or predictive value of it. You just need to select the imported datasets you would like to use for validating. You can use the default statistic configuration or modify the statistic parameters. For each validation that you execute, you can see different survival analysis results, including concordance index, log rank test, ROC curve, Kaplan-Meier, among others. You can view the assigned clusters to each sample, and also copy the executed R script to import in your R environment. You can optimize your gene signature for keeping the genes which are contributing to the prognosis. Bioplat offers different feature selection algorithms and meta-heuristics. Considering that you will train a model, you should use a training, a testing, and validation datasets for avoiding overfitting. You can use 3 different Bioplat datasets, or you can split randomly one of them. We will split the bandbigger one. The blind search will look for the best solution in the complete search space. However, you can reduce the solution space, restricting the number of genes for the solution. In this example, we will look at a subset of the original gene signature with 46 or 47 genes. In the blind search result view, you see the best in training, but most important, we can also see the result in testing and validation datasets. You can also open any of the results as a new potential gene signature to work with. Instead of using a blind search, you can use the particle swarm optimization meta-heuristic for reducing the solution space in a smarter way. You configure some PCO parameters. When finished, in the particle swarm optimization result view, you see the best in training, but most important, you can also see the result in testing and validation. You can also open one of the results.